All right, so we talked about the primary structure of proteins as well as the secondary structures in proteins. Now we're going to talk about what a tertiary and a quaternary structure is. Okay, so this is actually an example of hemoglobin. This is the enzyme in your body that carries oxygen as in the red blood cells. It actually consists of 4 different polypeptide chains. Each one is folded onto itself, and then those 4 are arranged in this shape here. All right, so what do we mean by tertiary structure? This is the overall 3-dimensional shape of protein that's formed between either attractive or repulsive interactions between the amino acid residues. Each protein has a unique tertiary structure. A lot of research goes into trying to figure out, given the structure of a protein, what its function should be. And those functions are always dependent on this fold of the tertiary structure. Okay, so it's going to be unique per protein. Unlike before, right? Notice that this particular protein has a bunch of alpha helices. A lot of proteins have alpha helices, but they will have unique tertiary structures from protein to protein. All right, so the forces that are going to be holding, right? So again, just kind of think about it. The cell has synthesized this chain of polypeptides. It's just linked one after the other, after the other, after the other, in a particular pattern. That's the primary structure. Those go ahead and fold a little bit, rearrange themselves to form these secondary structures, and then that further will wrap around and fold onto itself to form these unique tertiary structures. And so there's these interactions between neighboring amino acids. So for example, this one here and whatever is right here, that's holding those 2 alpha helices in this case together, that are holding that tertiary structure of the protein together. So those different forces are what we're going to talk about. So the first one is hydrophobic interactions. Okay, so remember, hydrophobic in the world of proteins is synonymous with nonpolar. So if we have 2 R groups that are nonpolar, they're going to interact with one another to form hydrophobic interactions. These tend to take place at the interior of the protein, right? So the very middle of the protein. Remember, the protein is floating around in the cell. It's floating around in this aqueous, water-rich environment. Those nonpolar amino acids don't want to be touching the water. So instead, they'll fold in on themselves and they're sort of interact with one another and not interact with the water on the outside. Okay, so these tend to be, they take place between nonpolar R groups, and they tend to take place at the interior of the protein. The sort of opposite, the flip end of that coin would be the hydrophilic interactions, right? So we have these polar R groups. They will tend to want to be on the outside of the protein because those are the ones that will interact with the water molecules that are floating around in the cell. So serine was an example of a polar R group. That's going to tend to be found on the outer sort of surface of the protein, sticking out and interacting with water. Okay, they're called salt bridges. These will hold like 2 different elements of a protein together. So for example, if I have one alpha helix here and another alpha helix here, they might be held together in this region here by salt bonds, or salt bridges rather. A salt bridge takes place between a negatively charged or acidic amino acid and a positively charged or basic amino acid. These form a, remember, like charges attract, these form what are called a Coulombic interaction, or these favorable interactions between positive charges and negative charges. So we have the negative end of this amino acid here forming a salt bridge with the positive charge on this amino acid here. There are also just hydrogen bonds that can take place. So these are going to be weaker than salt bridges, but nonetheless, there's a ton of hydrogen bonds that hold proteins together. We talked about how hydrogen bonds were incredibly important in terms of holding these secondary structural features together. And those were hydrogen bonds that take place between the backbone elements, the backbone carbonyl group and the nitrogen with the hydrogen. We can also have hydrogen bonding between the R groups on our within our protein, right? So if we have serine that has this OH group, that can form a hydrogen bond with an oxygen that's in an amide for an as an example. So hydrogen bonds can also hold, and there's actually proteins are made up of a ton of hydrogen bonds. A lot of them take place between the backbone, but again, we can also have hydrogen bonding in the side chains as well. And then there's one very special type of bond that we did talk about before, but these take place specifically between only one type of amino acid, and those are cysteines. Two cysteines can react with one another to form a new covalent bond. So it makes this sort of special compared to the other forces that we were talking about. Those were all like intramolecular or intermolecular forces, forces that happen between neighboring molecules. This is actually a new covalent bond that's formed. And again, this is called a disulfide bond. And so this is quite a strong structural element that will hold proteins together. All right, so within a protein, this is a great example here. We have, this is our tertiary structure of our protein. First of all, we can see all these different secondary structural elements. We've got these alpha helices I just colored in orange, and we have these beta pleated sheets here that are that I just colored in pink, right? So those are those secondary structural elements that occur because of hydrogen bonding within the backbone of the protein. But now these different secondary structural elements are also held together by forces largely between the R groups of the amino acids. So in this case, we have these hydrophilic interactions keeping this particular side of the beta pleated sheet on the exterior of the protein. Because remember, these like to interact with water that's floating around the cell. And then on the flip side, we have these hydrophobic interactions that are occurring in the interior of the protein, right? So this would be kind of buried in the middle. We have our hydrogen bonding or salt bridges. So these remember, salt bridges are just very strong interactions between a positively charged amino acid and a negatively charged amino acid. Hydrogen bonding is slightly less strong, but takes place between polar R groups that are able to form hydrogen bonds. So OH groups, amide groups, stuff like that. And these would be holding these two structural elements, secondary structural elements, kind of side by side. And then again, lastly, we have our kind of unique disulfide bonds that are taking place specifically between cysteine residues, and those covalently bond these two secondary structural elements to one another. So all of these forces in total contribute to the overall 3D shape of the molecule. We have a way of experimentally determining the 3D shape of a protein. That's why if you Googled, hemoglobin or something like that, you could actually see what an actual hemoglobin looks like. Theoretically, if you only know what the amino acid chain is, what the primary sequence of a protein is, it's really hard to predict how it's going to fold. It's a big area of science that uses a lot of computers, and they're still not really good at it. And that's because you can see how complicated all these different interactions could be. There's like a million different interactions that are possible. You have your 20 different amino acids. Every single amino acid in a protein has an R group, and that can interact in one way, either favorably or unfavorably, with another amino acid. So it's really hard to say, here's this primary structure, what is the tertiary structure? But looking at a tertiary structure, we can see common features like these that are actually holding them together. All right, so this is a really good type of question. So let's go through this. What type of interaction would you expect between the R groups of the following amino acids in a tertiary structure? Okay, so phenylalanine and valine. So how would I go about solving this type of problem? Well, what I'm going to need to do is, first of all, I need to know what phenylalanine and valine look like, right? So I'm going to have to go to my constants page here. Here's phenylalanine and here's valine. And this is when being able to categorize your amino acids are really important. Both of these are non-polar amino acids, right? So I could see that because this has this big non-polar benzene ring that doesn't have any sort of polar functional group sticking off of it. And valine is just a alkyl chain. So both of these are non-polar R groups, which is another word for hydrophobic, which means that both of these R groups are going to want to be located on the interior of the protein. So these will form those hydrophobic interactions that are predominantly found on the interior of a protein. What about cysteine and cysteine? This is this very unique example where these two R groups can actually form a covalent bond with one another, known as a disulfide bond. Let's just do another one. What about lysine? Oops. And aspartic acid. So again, I'm going to have to go to my constants page here. Okay, between lysine and aspartic acid. Okay, so again, this is when being able to categorize your amino acids is really important. Remember, aspartic acid, this is one of those acidic functional groups. So actually, if I was going to draw the correct structure of aspartic acid at physiological pH, I would make sure to include a negative charge on my R group. Likewise, I can correct these here as well. All right, so that's going to be a negatively charged R group. And then lysine has an amine, so it's a basic amino acid, which means that at physiological pH, its R group has a positive charge. I can go ahead and correct these as well. All right, and so what type of interactions do positively charged amino acids make with negatively charged amino acids? These would be what are called salt bridges. Okay, so this is a good example problem, being able to take two amino acids and determine what type of interactions they would form with one another. Okay, quaternary structure. So again, how a polypeptide folds back on itself is a tertiary structure. A quaternary structure is found in proteins that are made up of more than one polypeptide chain. So there are some proteins out there that are just one polypeptide chain. Other proteins are multiple polypeptide chains. Each one has folded onto itself and made its tertiary structure. And then those different polypeptides, folded polypeptides, associate with one another to form a quaternary structure. Okay, so again, hemoglobin is an example of a protein that has a quaternary structure because each one of these, so this orange one here, this red one here, this other orange one here, and then this, oops, that's just orange again. This one here. These are all four different polypeptide chains. Actually, two of them are the same. There's two pairs, but doesn't matter. Each one has its own N terminus and C terminus. It's folded onto itself to create a tertiary structure, and then those four have come together and stuck together to make the quaternary structure of hemoglobin. Okay. The subunits are held together by those same sort of stabilizing interactions that we see with tertiary structures. So at the interface between, so at this like interface here between these two subunits, there's likely going to be some salt bridges or some hydrophobic interactions or some hydrophilic interactions, et cetera, some disulfide bonds, et cetera, holding those two polypeptide chains together. With hemoglobin in particular, there's actually this thing called a heme, which is embedded in the middle of each of those tertiary structures. It contains an iron atom, and that's actually what the oxygen molecule is bound to. That's unique to hemoglobin, though. Hemoglobin and myoglobin. So myoglobin is basically something very similar to hemoglobin in that they both carry oxygen, but myoglobin consists of just one subunit or one polypeptide chain, whereas hemoglobin has 4 of them combined together. So myoglobin, with its just one polypeptide chain, would not have a quaternary structure, whereas hemoglobin, because it's consisting of multiple polypeptide chains, would have a quaternary structure. And so this is, I think, probably the best summary of what the last 3 lectures have been. First, we learned about the primary structure. That's just the sequence of amino acids from N terminus to C terminus. What's the identity? Glycine, alanine, lysine, asparagine, et cetera, what is the building blocks of the particular polypeptide chain and how are they arranged? Then those will arrange themselves in the secondary structure. Those are held together by hydrogen bonds between the backbone elements of the polypeptide chain. And then those secondary structures continue to fold back on themselves to form a tertiary structure. And if you have a multiple subunit or multiple polypeptide protein, those different polypeptides will arrange themselves in what's called the quaternary structure.